Hello, everyone, and welcome to today's live broadcast, Detection of Circulating Tumor Biomarkers Using the FIREPLEX Technology Platform, presented by Daniel Pregobon, General Manager of Platform Innovation of Abcam Incorporation. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Abcam. For more information about our sponsor, please visit www.abcam.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Daniel Pregobon. I will now turn the presentation over to him.
All right, thank you so much for the introduction. My name is Daniel Pregobon. I'm the General Manager of Platform Innovation at Abcam. Today I'm going to be talking about the platform that my group has been developing over the past few years for biomarker detection, and specifically some of the applications around cancer. So as therapeutics become better and better for many diseases, specifically cancer, we're realizing that the onus is really placed now on early diagnosis of the disease. So a critical pain point in research in medicine is really the discovery and the validation of stable, reproducible biomarkers that come from minimally invasive samples like plasma, serum, saliva, or urine. Proteins are the traditional biomarkers, and specifically circulating proteins in plasma or serum have been used or attempted to be used for clinical screening of cancer. Some examples of this are CA-125 that have been used for ovarian cancer in women, and prostate serum antigen, which has been used for prostate cancer in men. And of course, these are single molecule biomarkers, and so theoretically they're the most easily interpreted. Now, the problem with this is that as the field progresses, we are learning that single protein biomarkers are really proving to be insufficient to describe very complex diseases like cancer. And as we learn more about disease, we're learning more about how the different analyte classes interplay to not only tell you what the current disease state is in a patient, but where the disease is going. So what we're learning now is that biomarker signatures that utilize multiple targets and, more importantly, look across multiple analyte classes will likely yield the most robust diagnostic indicators for future diagnosis. So microRNAs are, for the last 30 years, have become the emerging biomarkers. And in comparison to their protein counterparts, they're ideal in many ways. Specifically, they have short known sequences, and they're largely conserved across species. They actually regulate most of the genes in our bodies, and what's interesting about microRNAs is that a single microRNA, instead of regulating a single gene, can actually regulate tens or even hundreds of genes. They're highly stable in biofluids. They're also highly stable for storage reasons. So typically, microRNAs are coupled with proteins that protect them from RNase degradation, or they are hidden in membranes, in microvesicles or exosomes. An interesting aspect of microRNAs is that they have tissue-specific signatures. So if there's a tissue-specific indication or effect of a disease or a therapeutic, that can be detected via microRNAs. And as we'll talk about significantly today, there are disease-specific signatures, especially in cancer, where microRNAs really got their first look into diagnosis. So microRNAs have been published quite extensively in cancer and in cancer metastasis. When microRNAs were first discovered, the first diagnoses were done in solid tumors. where people would digest a tissue, extract the RNA, and look at differential expression of microRNAs in tumor versus healthy tissue. But as of about 2008, where we discovered that microRNAs are actually found in circulation, we realized that there is much more potential for a minimally invasive biofluid to be obtained and to diagnose cancer via circulating microRNAs. And so much of the focus today is really on microRNAs that are in circulation. Interestingly, not just for diagnosis, microRNAs are also being investigated for use in therapeutics or to monitor the response to various therapies. So certainly they are seeing quite a large spread use as far as cancer diagnosis and therapy. Not just with microRNAs, but with any biomarker, they all follow a pretty similar development pipeline, which is broken up into three main stages: discovery, qualification and verification, and lastly, validation and clinical implementation. The discovery phase can be akin to casting a very large net. Typically, researchers want to look at all of the possible biomarkers of interest, and they want to do this over a handful of different samples representing various cohorts. And the idea in the discovery phase is to do a very deep dive on a few samples to identify a few candidates that could be used in a biomarker signature. After discovery phase, what's typically yielded are 10 to maybe hundreds of different analyte candidates for use in diagnosis. And what's done in the qualification and verification stage is to use these targets, but to take a much broader dive into many, many different samples. So typically what we'll look at for qualification and verification are tens to hundreds of targets, but we'll do this over hundreds of samples. Finally, once we get the statistical rigor that we would need to take this any further and consider clinical implementation, we would then move into a validation phase, where now we have maybe narrowed down our target signature to 4 to maybe 10 analytes. And instead of looking at hundreds of samples, now we're going to look at much broader classes of samples. And we're specifically going to do this over many different sample types and many different patients to really get a snapshot of the heterogeneity that we typically see with patient cohorts. So again, we'll look at maybe 4 to 10 different analytes, and we'll do this over thousands of samples before we decide if this is clinically relevant. So now we'll take a little bit of a dive into looking at the FirePlex biomarker profiling platform that my team has been working on. The FirePlex platform was really designed to be a versatile multiplexing tool that allows researchers to perform multiplexing with instrumentation that they currently own. One of the nice things about the platform, it was specifically designed for detection directly from crude digests of biofluid and tissue. So we have developed our assay such that we do not require, for instance, microRNA purification or total RNA purification. We can simply digest a sample and then detect microRNAs directly in that sample. The assay also works with very complex sample types, including heparin plasma, exosomes, formalin-fixed tissues, and even dried blood spots, for example. We have a versatile platform in that we can perform multiplex detection of microRNAs, and we can also do detection of proteins. And it's versatile also in the fact that our assays are completely customizable, and that we work with researchers to build the exact panel that they need for their specific application. Again, it was designed specifically for use in biomarker discovery and verification. We work with researchers who are looking to do hundreds of microRNAs over a few samples, or those who are looking to take a deeper dive, tens of microRNAs into hundreds of samples. So we have really designed the platform to spread the entire biomarker development pipeline. Lastly, we know that capital equipment is somewhat of a constraint, especially in research settings. So we have designed the assay for readout on standard instrumentation. And the first system that we looked at was a standard flow cytometer. The platform is based on encoded hydrogel particles. And with these particles, we're able to do in-well multiplexing, typically up to 65 barcodes for microRNAs in a single well, or 75 barcodes for proteins. As shown in the middle of the slide, our particles are rod-shaped and have 3 functional zones. The 2 end regions of the particle, we put different levels of fluorescence in those regions, and those act as a barcode, which will then identify the analyte that is being quantified in the probe region that is in the center of the particle. Now, in every well, we'll have multiple copies of different particles with different barcodes that are detecting their corresponding analytes. So we can have robust measurement, again, of up to 65 microRNAs in a well or 75 proteins. An interesting thing about the platform is that we built this on inert hydrogel substrates. Now, this is very different to the glass counterpart that you would see in a microarray or polystyrene that you would see in a bead, because in a hydrogel we actually have binding in a three-dimensional volume. This gives us optimal thermodynamics, but also gives us a very large dynamic range for detection. And because the hydrogel is bioinert, this also allows us to detect directly from crude biofluids without the need to do any blocking step in BSA or anything else. Again, we have a very versatile chemistry that we can apply to the particle, and this really allows us to adapt the particles to the specific need and to use them across different target classes. And again, as we already mentioned, we perform readout on a standard flow cytometer. And the way this is done is that our rod shape will flow past the detection zone of a cytometer, which would then think that the rod is actually comprised of 3 closely spaced cells that are going by the interrogation region. So each one of our particles that goes through a flow cytometer is detected as 3 events that is later decoded using our software. So to have a quick look at what the software offering is, we provide instant bioinformatics with our Fireplex Analysis Workbench. Now, this is a Java-based piece of software that really runs anywhere. It has simple point-and-click GUI, and it doesn't require any scripts. This will allow you to lay out your experiment, be it across multiple targets and multiple plates. It will allow you to easily import those data for quick interpretation and analysis. We allow a broad range of visualization from heat maps to bar plots to volcano plots, and also provide statistical information as far as ANOVA analysis across cohorts. We talk about p-values, and we also provide corrected p-values that take into account the multiplexing that is performed in these assays. All of the images are publication ready, and they can be scaled to meet your needs. Another really interesting piece of software that we're developing, and this is really for researchers who are unsure of which targets they want to quantify in their multiplex assay. We have the Fireplex Discovery Engine. And what this allows researchers to do is to type in their field of interest, and with the click of a button, what the software does, it will actually analyze the most recent publications in that area to provide a word cloud of the microRNAs that have been most recently published. It will provide the genes of interest that are also published in those papers, and they'll also provide you a list of authors that are actively publishing in the area. And all of these microRNAs and keywords are clickable, such that you can dig in deeper and see how those microRNAs were referenced in the various papers, or to see an annotated list of the publications. So I encourage you to check that out. Now we'll talk a little bit more about the nuts and bolts of, we have a platform, but how do we actually use the platform to quantify microRNAs and proteins? This is a snapshot of the protocol that we use for microRNA detection, and it's really broken into 4 main stages. In the first stage, we will hybridize microRNAs in a crude digest of a sample with our microparticles in the well of a microtiter plate. We will then perform a labeling step. We will then elute off the labeled microRNAs and amplify it via universal PCR. We'll then transfer the amplicon back onto our particles, where we will hybridize, add a reporter for fluorescence, and scan them on, again, a standard flow cytometer. And lastly, as I mentioned, these flow cytometer files can be loaded into our software for instantaneous analysis. Now we'll take a look at the molecular workflow. In the probe region of our microRNA detection particles, we have an oligonucleotide probe that has 3 regions on it. The center region of the probe is specific for a given microRNA target, and the 2 end regions are specific for universal adapters that will then be ligated onto the microRNAs. So in the first step, again, we are capturing a specific microRNA onto each specific particle that is floating in the microwell. We will then ligate these universal adapters onto the ends of the microRNAs. And again, it's important to note that for each microRNA, these adapters are the same, such that once we elute off those labeled microRNAs, we can perform an amplification with the same primer pair. And this was really designed to try and avoid any sequence bias that you might see in typical PCR, where you would have a different set of primers for each microRNA that's being amplified. After we've amplified the sample, we will then rehybridize it back onto the same particles that were used for the initial capture. And again, this provides 2 layers of specificity in that we have 2 unique hybridization steps to ensure that we are hybridizing the correct target to the correct particle before readout. After we've hybridized those biatenylated amplicons, we will then label with a fluorescent reporter again, and we will read out on a standard flow cytometer. Now, in order to assess the sensitivity of the platform, I always think it's most useful to look at the amount of sample that is required for detection in a given modality. So the way we looked at this, we took 2 reference panels to detect microRNAs either in crude serum or in total RNA that was purified from a tissue sample. So what we look at for crude serum, we looked at an input of 40 microliters down to approximately 5 microliters of input. And the targets that we were assessing here were either very abundant, moderately abundant, or lowly abundant in those samples. And what we see is that when dropping off from 40 to 20 to 10 microliters of serum, we're still robustly detecting most of the targets in our reference panel. Again, showing very highly sensitive detection of microRNAs, which are already very rare in serum samples to begin with. Now, in terms of total RNA, while most platforms typically look at microgram or certainly nanogram levels of input for their assay, we are measuring our input here in terms of picograms. So we started off with 2.5 nanograms and did dilutions down all the way down to 4 picograms. And what we're showing here is robust detection of a good number of targets all the way down to just tens of picograms of total RNA that was input. So again, this just goes to show that even though we're detecting in crude samples, we still have a very highly sensitive assay. Another interesting aspect of the platform is that unlike traditional qPCR, our system is intolerant to PCR inhibitors. One that always comes up is heparin. So what we did is, using the same patients, we did a single blood draw and collected into multiple different tubes where we had K2-EDTA, sodium citrate, or sodium heparin. And what we're showing here is that regardless of the anticoagulant or the preservative that we used, we're able to show very robust detection of all the targets and very, very strong correlation across the different collection methods. So again, this just goes to show that the assay, because we are washing away any remnants of the sample before we move on to the labeling and detection step, we are very robust and resistant to traditional issues that you might see with standard qPCR. When first launching the platform, it was very important for us to do a platform comparison, where we have found that a lot of the researchers that we work with are actually using multiple platforms to do orthogonal studies, just to show validation across multiple platforms to really take that piece out of the equation. So what we did is we used some of the most common platforms that we see our collaborators working with, and those include qPCR and also Illumina sequencing. We looked at 3 different types of tissue, and we purified RNA from brain, lung, and liver. And what we did is we measured the fold change of each target relative to the average of that target across the given samples, and then used these fold changes to compare platform to platform. Now, something interesting to note here is that the sequencing, in order to perform that study, we had to use 1 microgram of input RNA. For qPCR using the tilde cards, we had to input 60 nanograms. And with our platform, we were able to use only 5 nanograms. But as shown on previous slides, we very well could have gone much lower than that. And even with these very, very small amount of total RNA input, we're still showing excellent correlation with both sequencing and qPCR. And we typically see Pearsons above 0.9. Our microRNA offering, again, is really designed to address all of the biomarker development pipeline. We have developed discovery panels that will allow researchers to look at about 400 circulating microRNAs. These were all selected from peer-reviewed studies. We also have customizable discovery panels for rodent, immunology, or plasma and serum markers. Now, for more moderate panel that might be used in something like verification, we have focus panels. And these are comprised of 65 microRNAs that have been selected for multiple research areas, including tissue toxicity, oncology, immunology, cardiology, and neurology. Lastly, for the researchers who are studying novel markers or combinations thereof, we do allow for the complete customization of panels. Any microRNA that's annotated in MERBase, we can build a custom panel and turn that around very rapidly. Again, it can be comprised of anywhere from 5 to up to 65 microRNAs in a single panel. And just briefly, because we have talked about the importance of looking at multiple analytes to diagnose disease, we'll look into our multiplexed immunoassays. Now, these are performed as a standard sandwich assay, where we'll have a capture antibody that is attached in the probe region of a given barcoded hydrogel particle. We'll then capture a target, and we'll include a detection antibody that has a fluorescent moiety on it for detection once a sandwich is built on a particle. Again, all of these assays can be read out on a standard flow cytometer. They are all built in a pick and mix format, meaning we have a broad variety of content that can be included in the platform, and researchers are able to then pick and mix which targets are most relevant to their research. An important thing to note that distinguishes our immunoassays from most others is that as Abcam, we are very good at making our own antibodies. And because of that, we are building rabbit monoclonal antibodies, and we're predominantly using these to build our multiplexed immunoassays. And the rabbit monoclonals have really been shown to provide the best sensitivity and specificity possible for these types of studies. And just to show a little comparison of our multiplexed immunoassay versus other suppliers on bead-based assays, we're showing here for the 15 most important human cytokines, we typically see a much broader dynamic range. And in all cases, we're seeing very good sensitivity compared to other suppliers. Similar to our microRNA assays, we do offer some pre-built tools and also some custom tools for researchers to do the type of study that they need. For instance, we provide specific panels for cytokine release syndrome. We also have a panel for immuno-oncology. We have panels for toxicity. And again, we allow the customization by pick and mix of any of the content that we currently offer in our portfolio. So now that we've looked at the platform and the assays that we've put on the platform, what we did is we're going to summarize a couple of the research studies that have been performed using the system. And again, these are in the area of cancer, where the first one we'll look at was performed by a Swiss group where they were looking at non-small cell lung cancer. And they were looking to see if they can identify a signature of microRNAs that could be indicative of the overall survival of these patients. So I'm sure this audience knows quite a bit about lung cancer, but just to point out a few things, lung cancer accounts for about a third of all cancer-related deaths. And with a 5-year survival rate of only 2%, it's a pretty difficult disease to deal with. Now, chemotherapy is known to improve clinical outcome of non-small cell lung cancer, but as is the case with many cancers, it's really dependent on the correct disease classification and patient stratification. There have been some previous studies that have indicated that microRNA signatures could potentially be used to classify and prognose biomarkers, be prognostic biomarkers of non-small cell lung cancer. And so the aim of this study was to really dig deeper into that and to perform a multi-site prospective clinical trial with the main goal of really identifying circulating microRNAs that can be used as prognostic markers of overall survival. Now, alongside this, there were a couple other goals of the study, and they were to identify microRNA signatures that were potentially related to disease stabilization. They looked at microRNAs that could be indicative of tumor shrinkage or the time to disease progression following chemotherapy. So just very briefly, these studies were performed using PAXgene blood. Again, they looked at 50 patients, and they monitored those patients over time. And of the key findings in this study, what they did is a traditional biomarker development where they looked at 1,347 microRNAs in a discovery phase, and from those they identified 10 microRNAs that were associated with overall survival. and most significantly of those was microRNA 29A. And as shown in the Kaplan-Meier plot to the right, those patients that had a very high expression of miR29A in circulation actually had the poorest outcome. Secondly, they found 12 microRNAs that were associated with tumor shrinkage, and of those, the most significant was microRNA 665. As shown in the plot here, with the higher expression level of that microRNA, they actually saw a greater percentage of tumor shrinkage. Lastly, they found 10 microRNAs that were associated with the time to disease progression. And again, the main player here seemed to be microRNA 223. And again, higher levels of miR223 corresponded to the poorest outcomes for those patients. So it's interesting here that we're looking at three very different measurements, and of those measurements, we're finding 3 microRNAs that seem to be the key players in each. Now, the next study that we looked at was a little bit different. Instead of just diagnosing the disease, this is more of a mechanistic view of the microRNA 200 family, and specifically the role in metastasis and how extracellular vesicles play a role in this whole system. So metastasis, in this case they were looking at breast cancer, where metastasis is the major cause of mortality. Previously, research has shown that the microRNA 200 family, which includes 200A, B, C, 429, and miR141, have been shown to regulate the epithelial to mesenchymal transition, which is obviously a very key step in cancer spread. So tumor cells are known to actually secrete a large amount of extracellular vesicles. And again, depending on their size and their place of origin, these are referred to as exosomes, ectosomes, microparticles, etc. But here we'll just broadly call them extracellular vesicles. So tumor cells are known to secrete a very large amount of these extracellular vesicles, which contain all sorts of biomolecules, including proteins, messenger RNAs, microRNAs, etc. So even though there have been hypotheses, it's never been shown that extracellular vesicles can confer metastatic properties to cells that would otherwise be poorly metastatic in vivo. So with no surprise, the main objective of this study was to see if that was the case. Can we determine if microRNAs transferred by extracellular vesicles do indeed confer metastatic properties on cells that would otherwise be poorly metastatic? The research was actually broken into a series of multiple experimental chunks where they look at some of the mechanisms both in vitro and in vivo. And in this case, they built a very comprehensive study to prove their points. They performed several fundamental experiments to really drive to the conclusion that these observations are directly related to the microRNA 200 family, and that they're also related to those family members that are packaged in extracellular vesicles. So for example, they looked at some knockouts where the miR200 family was not expressed to see if the results were the same. They also looked at RNA digestion of samples to ensure that what they were looking at were actually microRNAs that were packaged in membranes and not those that were free-floating in solution. So again, they did a very comprehensive study to demonstrate their key results. Now, some of the key findings of this study were that the microRNA 200 family is in fact elevated in metastatic cancer cell lines. So in this case, we're looking at mouse cell lines from a tumor, and we have 4T1 cells that are very metastatic, 4T07 and 67NR cells that are not that metastatic. And what we're showing in this heat map are that the 4T1 cells are actually expressing a much higher level of the miR200 family compared to the non-metastatic cell counterparts. Another interesting finding is that the extracellular vesicles that are being secreted from those cells also show higher levels of miR200. Now, this is interesting that the results specifically state that the microRNAs in the extracellular vesicles, in this case, are very similar to the abundance of those that are in the cell lines themselves. So there does not seem to be any preferential packaging of microRNAs into the extracellular vesicles, according to this study. Next, what they showed is that poorly metastatic cells, those that are co-incubated with metastatic cells, will actually take on the metastatic properties of those metastatic cells. So they did this with a couple different experiments where they either co-incubated in a homogeneous format or they looked at a transwell format where the cells were not in contact. And in both cases, they were able to show that the non-metastatic cells took on the metastatic properties of those metastatic cells. And again, the hypothesis is that this is accomplished by the transfer of this microRNA 200 family via extracellular vesicles. So then moving into the in vivo studies, what they did is they had several mice with various cancers, and they showed that the mice that had the metastatic tumors actually did show an elevated microRNA 200 family level in circulation. And lastly, and I think most interestingly, in mouse and human xenograft models, they were showing that metastatic cell lines, again, with high MER200 family expression, were able to transfer the metastatic properties to nearby cells, but also to very distant sites. Again, really supporting this notion that the microRNA 200 family transferred by extracellular vesicles can be a major cause of cancer metastasis in living models. So again, I encourage you to look at both of these studies. I think they did a really good job of utilizing the platform and developing biomarkers to show very specific outcomes. So I encourage you to have a look and have a deeper read for some of the specific studies done therein. Now with that, I just want to summarize and again kind of highlight the uses of the FirePlex platform and really the most appropriate settings where the FirePlex platform was designed to work with a very limited sample volume. Again, with only 20 microliters as a standard input, we can typically detect 65 microRNAs. With only 12.5 microliters input for proteins, we can detect up to 75 barcodes in that case. Also, we work very well with crude samples. We can detect microRNAs directly from crude digests of biofluid. We can also work with very difficult samples like heparin, FFPE, CSF, and we demonstrated here detection in exosomes. Again, pointing to biomarker development, we work in high throughput settings or high plex settings. For those researchers who are looking to do discovery, to cast a very broad net over a few samples, we do that very well. But we were really designed for these high throughput assays where we're looking at hundreds or thousands of samples, and we want to do that over tens of targets. We have built a whole software suite that really helps streamline the bioinformatic and statistical analysis. And lastly, as I said before, this platform is really designed to be fully customizable. So if you have a study that looks at 10 proteins or 1,000 microRNAs or some combination thereof, the FirePlex platform was really put in place to help you. And with that, I thank you for your time and I open it up to any questions.
Thank you, Dr. Pragabond, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the recommended input of biofluid that you use for the miRNA assay?
This is a really good question, and it typically depends on if we're performing a discovery study or if we're doing a more focused study. Nominally, what we recommend as an input is 40 microliters of your biofluid, and this is then digested with an additional 40 microliters of our digest buffer. And with this cumulative 80 microliters, what we can actually do is perform a triplicate analysis for most biofluids. Now, in the case of our discovery panel, where we're looking at maybe 400 targets instead of just 65, we typically recommend a higher starting amount, and this has a lot to do with the fact that it's offered currently as a service. We want to make sure the samples are stable when being shipped. So again, we ask for a larger volume such that in handling these samples, we do have enough to input into the assay. But again, nominally, really depending on the sample, we typically recommend anywhere from 40 microliters, maybe even down to 10 microliters of biofluid.
Thank you for that answer. Our next question is, what type of cytometer does your platform work with?
Another good question, and again, the platform was made to be as versatile as possible. So we have tested over a broad number of flow cytometers, and the minimum requirement really is that the system we prefer to have a single blue laser and 3 colors for detectors, including green, yellow, and red. But we have worked with a number of Becton-Dickinson cytometers. We've worked with Beckman-Coulter, the Accuray, even the Millipore Guava system all work very well with our platform. So we encourage you, if you have a question about a specific cytometer, please get in touch with us and we'll tell you about the compatibility studies that have been done.
I would like to once again thank Dr. Daniel Pragabon for his presentation. I would also like to thank Abcam for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.